Thank you, Joel. Just a note to all of you, Actigard, also known as Bion. You might have heard the names used interchangeably. The name they're going to use in New Zealand for sales is Actigard. It's going through ACVM approval at the moment, and we're confident, hopeful, confident that it's going to be available later in October. That's the date that's been given for approval or otherwise, but that's what we're targeting it right at the moment. Our final speaker is Michael Franks, one half of Taskforce Green. Michael is CEO of Seeka, as you know, and Michael's going to start this presentation. I think he's going to use a couple of his colleagues at the same time, so welcome, Michael.
Thanks, Dave. This is going to be one of those sessions that either goes really well or a total disaster. I'm lucky enough today that we've got Professor Russell Poulter here. So we're flying him up from Otago because we've been distracted doing other things, as you can imagine, and dealing with PSA itself. And so we really haven't had that much opportunity to get ourselves properly prepared. But we're lucky to have talent like Russell here and willing and working for us, I guess. We also had Saray Avery here on Friday, and we've got Jerome Demmer here somewhere in the audience. Look, just to remind you before we go to these photos, which we're going to show you, but leave them there. Taskforce Green, we only really commissioned this thing on the 10th of June. It is a commercial research initiative, a collaboration, if you like. It's two sort of people who don't normally get together. We decided to collaborate. It's ourselves and EastPac. It's probably the only thing we can collaborate on. It's a fact. And essentially we decided to get together. Now, we've done a whole lot of things. A lot of the things that we've done since the 10th of June haven't worked. But we've got a lot of learnings. I mean, we've got our own lab now right up to speed and as good as any other lab operating. We have tried, under the guidance, and Murray Judd really heads up everything we do, we've tried to, if you like, inject antibiotics, just as you've just seen, into heavily infected plants to see whether or not combinations of cocktails of antibiotics might be able to act as a curative. We now know, with the wisdom of hindsight, and not that much further on, that we're trying to cure, it's like trying to cure leprosy. We're sort of like, we now know that those trials largely haven't worked and we're removing that orchard. We've now got very large scale trials underway where we're trying to use injected antibiotics as a preventative. These are big scale operations with trials that have all been approved, they've gone through the approval process, and we have injected plants to see whether or not we can actually stop or slow the spread of PSAV by injecting antibiotics in the field. Also, we've got approval in place now for us to trial products, just the same as Joel was talking about, whereby we can take plants, treat them with a product, then knowingly expose them to PSAV and see what happens, and that will start next week. And I thank all the neighbours who actually have given us their approval as part of the approval process of getting the people willing to say yes. And so, you know, there's a big effort going on at the moment. We don't have any magic cures, but it is happening. One other thing that we are just putting our minds to is how do we cleanse pollen? You know, it might be one thing to know how the pollen moves through the hive, but we've got a whole stock of pollen around. We know now how to test pollen. We just need to figure out how to cleanse it. And some interesting work's going on there. Now, before I ask Russell to speak, Clint's at the back of the room. He was going to get up here with me with a little pointer and point out some of these photographs we're going to show you. But one of the great things about Clint, not only is he dedicated to the task, he's also colourblind. And so when he gets the pointer out and points it up there, he actually can't see the damn thing. And so he needs to be down the back. And so he's going to do this little bit. These photographs have come from some work that has been done for us by Scion, by a guy called Lloyd Donaldson. I used to work with Lloyd quite a while ago, actually. I helped him set up and fund the confocal microscopy capability that they've got there. They've done this for us on a special deal. No one else could get this for this money. And so Clint from the back is just going to talk you through some of the images, only 4 or 5 of them. And after that, I'll introduce Russell Poulter. So off you go, Clint.
Thank you very much. Yes, these images are courtesy of Lloyd Donaldson at Scion. We originally contracted him to have an initial preliminary look at some plant material that was infected. We were interested, if possible, at the initial look just to find out exactly where the PSA was as far as where it was located, what cell structures were involved, if it was in the xylem, could we see visually how it was moving through, in, or around the xylem tissue. And it was very preliminary work. And with most preliminary work, it actually brought forward more questions than necessarily answers. But the images are quite stunning that he was able to produce. This image here in particular is all the wood that was given to him was infected Hort 16A canes. You can see here, these are xylem vessels that are surrounded by parenchyma cells and other plant features. But the initial target in this photo was this cloudy area, which we didn't originally understand if it was bacteria or not. And he used an actiflavin stain and to stain all this material, so that's why you see the spectacular colors. One of the questions that came up was the blue fluorescence that we were seeing in a lot of these cell structures. He was unable to identify it. It was something he had not seen himself before, and he thought that perhaps it would be worth looking at some control vines under the similar stain to see if it is in fact PSA related. But to zoom in further on that image and that xylem vessel, we could see that there was this cloudy structure here, which we could not conclusively tell if it was PSA in fact. Moving on. This is a longitudinal cut of a xylem vessel, and something of interest here is just going forward in research how we can understand. We understand that the wilt that we see out in the orchards is the effect of the bacteria clogging the vascular system, and we just want to understand what the bacteria is doing in the xylem vessels to create that inclusion. And then this image here, Lloyd was actually able to find the PSA. And so in this image, we can see our enemy. It is this rod-shaped structure here. This is loaded with bacteria. Here we have a xylem vessel, and of interest here is that this is not actually occupying a cell per se. This is actually occupying the intercellular space between cells. So here, one more. Sorry about that. The cells are actually confined here by the cell walls. This dark area is actually cytoplasm. That's a nucleus. So this is actually an area between cells. And so moving forward in research is to understand how the bacteria is actually moving through this intercellular space. And you can see more of the bacteria here occupying the intercellular space. So there has to be a mechanism of how it's actually traveling, whether it's an enzyme or somehow it's degrading cell walls to travel through the plant. And the last photo here shows a large quantity of bacteria occupying a xylem. So we can see obviously that it's thriving here in the vascular system. This large rod-shaped structure is actually a fungal hypha. That's sort of a secondary infection. This was exciting for us because of this area over here where we can see bacteria is actually somehow going from the intercellular space to the xylem. And so this indicates that there is some sort of degradation or breakdown of... It's all quite preliminary, but if we can understand how that bacteria got through that area through further research, then we can better understand how this bacteria is traveling through the vine, how it's causing its virulence, and then if there's enzymes involved with this, the genomic research can possibly target specifically those enzymatic functions, and that would be very useful for the screening process. And that's the conclusion of the images.
I mean, while those photos might be visually pleasing, anyone who finds them interesting probably just needs to get out a bit more, I think. But that's sort of like an indication of some of the work that's going on. One of the areas that we have been focusing on, and there's heaps, I mean, I could talk about a whole range of things that's going on, but one of the areas of work that is going on is the work around the genomic sequence. Look, I'm an accountant. I'm not a scientist. Before people started talking about the genomic sequence, I had no idea what we were talking about. We're lucky they've got really smart people like Russell Poulter here who understands it. And I think we're really lucky that we have this capability within New Zealand that we're able to tap into and to get work done very, very quickly and actually quite economically as well. This work, it's a national resource for us. It's been very useful. And Russell, in his own style, will get up now and present it. He wouldn't let me see his presentation before he came, but that's fantastic. The Taskforce Green Network is functioning well, and it's functioning well because we've got people like Russell Poulter in it. So you're really here to listen to him, not me.